Well, thank you, folks, for joining. My name is Mike Mancini. I've been running the GCC Center for Advanced Microscopy and Image Informatics for many years now. We've sort of been winding it down with the grant funding probably later this next year. We've got two talks today. One, Mike Bolt, as you see here on the slide, and Melody Bona. And let me go to the next slide, Mike. The two talks, sort of picked them for a variety of reasons, both good science, of course. Mike's is more of a phenotypic library screening project, connected more to the drug screening aficionados in the audience. And Melody's Bona will be coming in second. She's definitely more of the basic nuclear cell biology. But that project, much like many of the projects that we've taken on during the past many years in our center, sometimes assay development turns into screens, sometimes more just basic cell biology projects. So a little bit of both, but certainly her project is amenable to phenotypic screening as well. And at the end of each talk, please hold questions. We're going to have a little roundtable at the end. So with that, last comment, we're still in business for a while longer. And if there are people that are interested in some project ideas that we can help with, just send me an email and we can discuss. And with that, introducing Mike Bolt. Mike actually got a PhD from Baylor in the cell biology department a little over 10 years ago out of my lab. Went on a postdoc up in Chicago for 5 years, came back, worked at the Texas A&M Institute of IBT, and was a project manager for many projects as part of our center, and has in the last 6 months or so been back over at Baylor and the technical director of the imaging resource that we have here. And with that, I'll let Mike take it away.
Thanks, Mike. So today I'm going to discuss phenotypic screening and how we've used it to identify xanthomumol and other flavonoids as killers of bladder cancer. I'm also going to first discuss what is phenotypic screening, and a paper we had published recently I'll quickly go over about a Python program that we created in the lab for quicker analysis of these types of data sets. There is no I in science. There are a lot of people who've attributed to this work, so I want to make sure and show them off from the Mancini lab, both over here at Baylor and at Texas A&M. And then the BCM ACE3D core and the Labate lab of U of H. So cell painting and phenotypic screening. The cell painting technique uses common fluorescent dyes to identify several subcellular spaces: nucleus, mitochondria, actin, cytoplasm, nucleoli within cells. The important part of this is that these dyes are all very commonplace and pretty cheap compared to, say, screening with antibodies. So you can use these dyes to get a lot of extra data out of, say, a general toxicity screen where maybe you're only going to stain with DAPI. Now you can stain with these other dyes, keeping the costs down, and get a lot of mechanism of action data or other types of data simultaneously. And so here on the right is just example images of DAPI, cyto14, which is one of the dyes that labels nucleoli and cytoplasm, phalloidin, which is a common actin dye, mitotracker, and then the merge. These are in bladder primary fibroblasts. And then you can see as we go into 5637, RT4, and UMUC3, which are all bladder cancer cell lines, there are steep phenotypic changes in between each of these cell lines. So normally, or I guess in the beginning, Ann Carpenter came out with the cell painting, and her group, the JUMP Consortium, has used for a long time a very robust cell profiler analysis. And so when we started these, the one problem with Cell Profiler is it takes a lot of time to run. And so if you're going to be doing screens or any other kind of large experiments with cell painting, if you don't have really, really good computational resources, using the Cell Profiler method can take weeks to image data sets. So our goal was to create also an open source Python-based cell painting analysis program. And we specifically made it for fast, reproducible, and accurate morphological analysis of cell populations. Importantly, that can be run on a standard workstation. Like most image analysis, we load the images, tweak the hyperparameters just to make sure we can get good segmentation. We get cell and nuclear masks. We generate nucleolar and mitochondrial masks, which is new. Actually, I think the new JUMP consortium method can do mitochondria, but it didn't previously. And then feature extraction and EMD-based quality control, and then get your measurements. So here's just the stains of cell paint and then the masks that come from each of them. And so from this, we can then do compound tests looking for different changes to the different cell types. So for instance, here are your DMSO cells. If you treat with AMG 900, you get these fractured nuclei and really dim cyto14 staining. If you treat with berberine, you get these fragmented, really hyper bright mitochondria in red. And then rotenone, you get these cells that have smaller cell bodies as the mitochondria dysfunction begins to happen. And importantly, you can use this to look at drug interactions in between cell lines. So here we have U2OS and a common bone cancer cell line, or osteosarcoma, and then PANC1, which is a pancreatic cancer cell line. And when you treat them with these PIK5 inhibitor or PI3 kinase inhibitor, in the U2S, you see these vacuoles appear in the cytoplasm, which you also see for the PIK5 inhibitor, but the PI3K inhibitor in PANC1 really just kills the cells. And so you can see differences between cells with drug treatments very easily with this method. And then this is another one, rotlerin, that has mitochondrial effects. And so you see dimmer mitochondria and a little bit of a loss of actin network. And so, as I said, the important thing that we wanted to get out of space was the speed, and running the same data sets on the same computer, the same software, or the same hardware. Space is about 8 times faster than the JUMP consortium cell profiler, while importantly not losing. These are just metrics of how well you can get mechanism of action data from all these different inhibitors and things like that. And so we perform better, or at least not worse than Cell Profiler, even being 8 times faster. And so this was all originally programmed by a couple of really good programmers, and it's been modified throughout over the past year or so. My claim to fame is I came up with the acronym SPACE, which is Swift Phenotypic Analysis of Cells. I'm really good at acronyms, so if anyone needs help with one, just give me an email. But during this time when this was being built, I was looking at a library that we purchased of flavonoid compounds, specifically looking for which of those flavonoids could be estrogenic. And so once that was done, it came to me that we could also use this library to find toxic flavonoids for bladder cancer cells. And so that's when we came up with this phenotypic screen to identify toxic flavonoids for bladder cancer. Bladder cancer accounts for essentially 4% of the cancers in the US. It's very gender biased towards men. Flavonoids are phytochemicals that have a variety of therapeutic effects. A lot of them are anti-inflammatory. A lot of them are estrogenic. and then some have been shown to act against cancer, and then there's a lot that are also have pesticide abilities. And the reason I thought maybe this was a good library to test is many of the flavonoids that we eat end up in the bladder, and some of them can end up in very high concentrations in the bladder. So maybe some people are already treating themselves by eating foods containing these flavonoids. So this is the basic scheme. We chose 3 cell lines, 5637, UMUC3, and RT4. And we chose them, if you look down here, they represent different tumor grades, different HER2 status, different mutational and genomic instability facets. And so we treated these cell lines with our flavonoid library, which contained 244 compounds, for 72 hours. We then robotically processed these plates and imaged them on our Yokogawa CV8000 microscope, ran them through space, and then found the toxic hits. And so here is a heat map of all 244 compounds, blue meaning fewer cells, red meaning more, or these are all Z-score normalized to DMSO. And we found this patch of compounds that were toxic to the cells. Now, flavopiridol and flavopiridol hydrochloride, not surprising, those are transcription killers. They're going to be toxic to cells over 3 days. Rotenone is also well known to be toxic. They actually treat rats with rotenone to induce a Parkinson's-like state. And so that's a Parkinson's model of a rat model of Parkinson's disease. But some of these other ones have not been known to be toxic to cancer cells before. So these are just some example images. This is your DMSO. This is P276. P276 is a very closely related to flavopiridol, but it has been used in some clinical trials. And then casticin and phenoxydiol, you get shrinky cells dividing weirdly, or dimmer mitochondria, in addition to the lack of cell number. And so next we wanted to look at both cell type and dose response changes, effects of the flavonoids. And so this is in 5637. We did a small dose response from 10 micromolar to 100 nanomolar. And looking across the cell line, so 5637 versus RT4 versus UMUC3, there are definite cell specific, like biocanon A really only affects the RT4s. Degwelin hits the RT4s and UMUCs harder than it does the 5637s. And that can be shown here. This is a longer dose response curve from 1 nanomolar up through 10 micromolar. And so these flavonoids do show dose and cell type specificity. As I said earlier, the real benefit of using cell painting is the mechanism of action information you can get from these toxic compounds. So when I went back and looked at the cell painting data, not taking into account now more than just cell or nuclear count, 3 of the compounds, P276, phenoxydiol, and casticin, showed heavy changes to DNA-based features. So the area of the nucleus, EFC ratio, which is a measure of circularity, the DAPI intensity itself, and the DAPI contrast itself, how smooth or rough is the DAPI signal. And 3 others showed changes to mitochondrial features, whether it be the mitochondrial area, the mitochondrial intensity, or the mitochondrial contrast. And so to confirm kind of the DAPI effects, I wanted to know if any of these compounds were inducing DNA damage in the bladder cancer cells. And we use cisplatin as a control, but lo and behold, the 3 that we saw the disrupted DAPI features for casein phenoxydiol, and P276 all significantly induce gamma H2AX staining in both the 5637 cells and the UMUC3 cells. The casein effect has already been known. That's a published effect, but the phenoxydiol wasn't well known. And like I said, P276 is very similar to flavopyridol, so you'd expect some DNA damage if you're disrupting transcription like that. So we went and we didn't really know how xanthihumol might be affecting the mitochondria. So we have a new instrument called the NanoLive, which is a refractive index based holotomography microscope that allows for live label-free imaging. And one of the things you can see really easily on the NanoLive is lipid droplets. And another one is mitochondria. So while we did this experiment, I was originally looking for disruption to mitochondria. What we actually saw is over time, the xanthihumol treated cells lost their lipid droplets. And so this is a blow up of, this is the full refractive index, and this is just thresholding the refractive index images to show just lipid droplets. We followed that up by doing basic Bodepie staining to see, can xanthihumol get rid of lipid droplets? And if you compare the DMSO to the xanthihumol, there aren't many lipid droplets left. And even at the lower dose, there's a nice dose response curve in the UMUC3 cells of xanthihumol getting rid of the lipid droplets. And this is also true in the 5637 cells. So xanthihumol is getting rid of lipid droplets. That's, the cells don't have any energy. That's probably why there are fewer cells. We next wanted to look at whether these flavonoids can kill bladder cancer cells in a more 3D cell culture. So we made 3D spheroids of a variety of different bladder cancer and primary bladder fibroblast cells. And so when we did our three main hits from the screen, casein, diguelin, and xanthihumol, none of these had a major effect on the area of the primary bladder sphere. Again, primary bladder fibroblast. So this is a cell line. It's not from actual bladder. 5637, all three were toxic, and xanthihumol erases the sphere. For the J82 cell line, both diguelin and xanthihumol were toxic. Xanthihumol made a smaller sphere, whereas diguelin started to completely dissociate. In RT4, only diguelin was toxic, and then with RT112, all three were toxic to varying levels. So these are toxic in 2D, they're toxic in 3D. What about in a more physiological system? And so for this, we utilized what's called a CAM model, a chicken chorioallantoic membrane model. If you don't know about this, it's a very cool model where you basically get a fertilized chicken egg, you break the top of it off, and so you can see into the yolk sac and you see the blood vessels and the embryo, and then you can actually implant tumor cells and they'll grow, and this allows for intravasation. It's a really nice assay. And so you can then add drugs on top of it and see if your drugs are affecting the vesicles, the blood vessels. Do your drugs block the growth of your tumors in this system, it's a nice physiological symptom in collaboration with the ACE 3D Core and Hugo Villanueva here at Baylor. And very nicely, if you look at xanthihumol treated CAM model versus the DMSO, so these things here are the tumor nests of the UMUC3 cells, and you find many more in the DMSO treated CAM eggs versus the xanthihumol. So there is a nice decrease in the tumorigenicity of the cells when they're treated with xanthihumol. And so I hope that I've shown you today that the use of high throughput imaging and phenotypic analysis by cell painting can provide not only tox data, but nice mechanism of action data to large screens. I talked about this screen, 244 compounds, 3 cell lines. This paper is out for review now, but in the meantime, we've done a much larger screen, a 1400 compound food as medicine screen across 8 different cell lines using this method to find more components that in food that are toxic to bladder cancer. Xanthocumol is our kind of unique hit, is a common component of hops and has been shown to be toxic to other cancer cell types, including breast, prostate, and pancreatic. However, when you take it, people take it, it doesn't have high bioavailability in blood and tissue. I think actually this is in mice. But following beer consumption, this is actually a cool German study, they did find high concentrations of xanthocumol in urine in beer drinkers in Germany. So it does make it to the bladder as xanthocumol. There's a metabolite of xanthocumol called isoxanthocumol that does not have the same effect. It's really the xanthocumol that is having the toxic effect towards bladder cancer. And there's actually a clinical trial going on right now, unrelated to bladder cancer, just to see if xanthocumol is safe to give to patients. That's the whole purpose. But it's a nice convergence there with kind of what we found. And so, yeah, so this is a very powerful technique. We have 12 screening libraries now, including an FDA-approved library, epigenetic library, kinase inhibitor library. So if you're interested in any of this kind of research with what you're doing, please email me or Mike, and we can have a chat about running these kinds of assays. Thank you for listening.
Okay, as mentioned, questions, if there are any, please hang on to them until after the next talk by Melody de Bona. Hello, Melody. Melody is postdoctoral fellow at Sloan Kettering, currently on the job market, flying around interviewing else here and there, and is lucky enough to join us. We're lucky enough to have her join us here. As I said at the beginning, which Melody didn't hear, Melody's is more of a basic science nuclear cell biology story that is very much amenable to the kind of phenotypic screening that Mike just talked about. And take it away, Melody.
Thank you for the introduction. Let me share my screen. Do you see it?
Yep.
And do you see my pointer?
Yes.
Amazing. All right. So as Mike told you, I'm more of a mechanistic biologist. So thank you for the opportunity to tell you a little bit more about my results during my postdoctoral research on micronuclear rupture. So let me start from the very beginning, right? What are these micronuclei? Micronuclei are the byproduct of chromosomal instability, which is this pervasive hallmark of cancer that is characterized by chromosome missegregation and is associated with metastasis and poor patient survival. And this is mostly due to the presence of micronuclei, which are their byproduct, right? So chromosomal instability is characterized by chromosome missegregation, which is basically problems in the division during mitosis. So for those of you who don't remember very well how mitosis works, basically just before its onset, we have the DNA in the nucleus that is duplicated. Then for the chromosomes to start to form, they align on this metaphase place, they get attached to this mitotic spindle that then pulls apart these two copies of DNA, so they end up actually at the opposite pole of the cells, and then the nucleus form, and we have the division to exactly identical cells. Sometimes this not happen, and especially in cancer and especially in chromosomal instability, there are a lot of errors in this process that can happen as soon as metaphase, when there is a formation of this pole. Instead of having two poles that pulls the chromosomes, there are more than one, a multipolar mitosis. And this is important because they never have the same strength. So basically, the chromosomes are never divided equally into the two daughter cells. And then we also have these figures that we are the ones that we are mostly using actually to score chromosomal instability during interphase. The first one that you see here is called a chromatin bridge, and is basically a chromosome that is attached to two ends of the mitotic spindle, so it's stretched in the middle. And this other one is what we call a lagging chromosome. As the name suggests, it lags behind, again, because it's not very well attached. What all of these things basically have in common is that they end up having like some DNA that is away from this big mass of chromosomes. So when the nucleus forms, it's not encapsulated in the main primary nucleus, but it gets encapsulated into its own micronucleus. And micronuclei are quite weird structures. As you can see from this movie, at some point you will see a white arrow appearing. Whatever it's inside the micronucleus keeps misaggregating and ends up in the new micronucleus every cell cycle. And this is important because micronuclei are driver of tumor progression. So whatever it's inside the micronucleus is not correctly maintained. Micronuclei have a lot of DNA damage, alteration in transcription, alteration in nuclear envelope, but what is even more important, micronuclei often undergo irreversible rupture, which is the most occurring event in the life of a micronucleus, and basically they are like ticking bombs in cancer cells, because when micronuclei rupture, it's truly like an explosion. Whatever it's inside, the chromosomes get fragmented into thousands of pieces that then they are aberrantly rearranged in a process that we call chromotrypsis. And as you can imagine, this leads to translocation, fusion proteins, mutations, so genomic heterogeneity, which is important for therapy resistance. But also micronuclei already have different epigenetic marks, and when they rupture, these differences get exacerbated and they become heritable. So again, genomic heterogeneity, but here we also have phenotypic heterogeneity that again is correlated to therapy resistance. And finally, when the micronucleus rupture, neither the DNA nor the cytosol are ready for this interaction because it's not during mitosis. So the DNA gets recognized by cytosolic sensing pathway that sense it as a viral signal, because the only other moment in which we have a DNA in the cytosol that is not from mitosis, it's from a viral infection. And what is important is that these pathways, when they are chronically activated, which is the case of cancer cells because these micronuclei keep rupturing, right? They shift, instead of having a pro-inflammatory effect and call the immune system, actually they shift towards a non-canonical response that is actually immunosuppressive and pro-metastatic. So I hope you can understand now how it is truly, truly fundamental to understand the mechanism of micronuclear rupture, because in that case, then we can stop all of this process to happen. But until now, it was not possible because we didn't know not only the mechanism, but also why micronuclei were ruptured, which were the causes. And several people have studied micronuclei, and they showed us that they're very, very different from the primary nucleus. Here I'm showing you two seminal papers in the field from David Pellman and Emily Hatch that show, for example, that micronuclei have defective import, and this import leads to have less levels of this lamin B, which is a structural protein. So they have a more fragile envelope. But this still doesn't explain why they rupture without any mechanical stimulus, and also why these ruptures are not repaired. So basically what I did during my postdoc was finally answering these questions, putting together all of these observations, and using a model that is based on a size-dependent organelle load. And the organelle in this case are mitochondria, because look at this micronucleus. It is rupture because it has lost the yellow nuclear integrity marker. And look at the pink mitochondria, how they are engulfing it. While on the other hand, this other one is intact because it's yellow, and you can see that the mitochondria are farther away. And indeed, by analyzing these super resolution images, I was able to draw a correlation between mitochondria proximity and micronuclear integrity. And following this micronuclei in live cell imaging, I actually saw that the micronuclei that rupture were the ones that were spending more time in contact with mitochondria, while micronuclei that were spending time away from mitochondria were not rupturing. So I started to hypothesize that maybe truly mitochondrial proximity was the cause of micronuclear rupture through the production of reactive oxygen species. To test this hypothesis, the first thing that I needed was a marker of rupture. So, like I used and validated several, here I'm showing you only the loss of lamin B1 because it was the most stringent one. When micronuclei lose lamin, they're completely collapsed. There is no membrane left. So you have on top an example of an intact micronucleus. So they have less lamin than the primary nucleus, but as you can see, it's visible, right? And below you have an example of a rupture micronucleus, which is completely devoid of lamin. And then I started to play with the level of ROS in the cells. So let me walk you through this graph. On the y-axis, I plotted the amount of rupture micronuclei. So those micronuclei completely devoid of lamin. On the x-axis, it is just different cell lines to show that is like a generic mechanism. In purple, I have treatments that increase the level of ROS. In this case, I use several, but here I'm showing you only the direct addition of hydrogen peroxide, which is the main species that leaks from mitochondria. And in green, treatments that reduce the level of ROS, so ROS scavengers. Again, I use several. Some of these are also specific for mitochondria, but this one that I'm showing you, it's N-acetylcysteine, which is a precursor of glutathione, which is a pancellular antioxidant. So, as you can see, if I increase the level of ROS in the cells, then I also increase the level of ruptured micronuclei. And if I decrease the physiological level of ROS, then I'm also able to reduce the basal level of ruptured micronuclei. So this means that ROS are the cause of micronuclear rupture because of mitochondrial proximity. So we found the causes. We are missing all of the mechanism. For this, we decided to focus on this escort-3 complex because it has been observed that rupture micronuclei, and actually micronuclear rupture has been proposed to be a function of its overzealous repair because it usually repairs the membrane. And it does so by forming on top of its scaffolding subunit CHIM7, but only when CHIM7 is bound to the inner nuclear envelope through its binding partner LAMD2. CHIM7 is very small and can enter the nucleus through passive diffusion through the nuclear pore complex. So to prevent this interaction when there is no need for it, usually CHIM7 is exported by this exportin that is called XPO1. When there is a rupture, as you can see here, basically there is a hole that is bigger than just the nuclear pore complex. So the flux of CHIM7 is so much that saturates XPO1. So CHIM7 is able to stay in the nucleus long enough to bind LAMD2, catalyze the formation of the complex that seals the membrane. So we wanted to know whether this complex was involved in rupture, and we decided to knock it out. So we knock out CHIM7, the scaffolding protein, and other two essential proteins of the escort-3 complex. So basically, without any of these proteins, the complex cannot work. As you can see, by knocking out CHIM7, we are able to reduce micronuclear rupture independently on the level of ROS. So this means that CHIM7 is involved in this mechanism. What we were very, very surprised to see was that this was not true for the other subunit of the escort-3 complex. So this means that the escort-3 is not involved in rupture. What we're seeing here is an escort-3 independent role for CHIM7, which instead is necessary for micronuclear rupture. Not only this, but we also saw that CHIM7 somehow gets stuck into the micronuclei, forming these big blobs that you can see here in IF that are completely dependent on ROS and mostly happening in intact micronuclei. Because here, instead of like plotting rupture micronuclei, I plotted micronuclei that have these blobs of CHIM7, and I distinguish them between lamin B positive or lamin B negative, intact and rupture. And as you can see, if I increase ROS, I increase the number of micronuclei with these blobs of CHIM7, and they're mostly happening in micronuclei with lamin. So this suggests that somehow ROS is increasing the accumulation of CHIM7 in micronuclei, and this precedes rupture. So I thought that maybe this was my mechanism somehow, right? So the first thing that I asked was how ROS were able to increase this accumulation of CHIM7 in micronuclei. And I told you before that CHIM7 usually is exported by XPO1, but I found in literature evidences that ROS inhibit XPO1. So I thought, oh, maybe this is my mechanism. First of all, how ROS inhibit XPO1. They do so by increasing the oglucnacylation of nucleoporin, which are the proteins that form the nuclear pore complex. So basically, they stick a molecule of sugar to these proteins, and XPO1 binds with more affinity to this sugar. So instead of transiently associating with proteins of the nuclear pore complex and going in and out and do its work, basically it gets stuck at the nuclear pore complex, and then it cannot go into the cytoplasm and release its cargo. So the first thing was actually checking whether there was more oglucnacylation under ROS condition. And to do so, I analyzed the fluorescence images, and I saw that indeed there was more oglucnat signal under ROS condition. And then using cells that express XPO fused to a fluorescent protein, I actually saw that somehow XPO was stuck at the sites of nuclear pore complex in micronuclei. So we decided to follow these micronuclei over time. Again, in cells that express XPO fused to a fluorescent protein in green, and in red, a marker of rupture that is lost as soon as the micronucleus is ruptured. So we are able to pinpoint in time when this event occurs. And as you can see, in the micronuclei that do rupture, all of them in the hours preceding these events, they are accumulating XPO1 at their nuclear rim. So to make sure that this was truly the cause of rupture, we decided to mimic what we are seeing with ROS by pharmacologically inhibiting XPO1. And we used selinexor, which is it's a specific inhibitor that is also used in clinic already. So as you can see in these cells, if we reduce the level of ROS, we reduce rupture, cool. But if we inhibit XPO1, we are able to increase micronuclear rupture, and further co-treatment with ROS scavenger is not able to rescue this rupture anymore. So this means that ROS are rupturing micronuclei through nuclear export inhibition, and specifically of CHIM7, because if we knock out CHIM7, then none of this treatment have anymore any effect. And to be completely sure that it was CHIM7 specific, we decided to use mutants that are unexportable. So we knock out the CHIM7 from the cells, and then we reintroduce the CHIM7 that we wanted. In this case, I'm showing you only one mutant with point mutations in the nuclear export sequence that makes it unrecognizable by XPO. So as you can see, by using these mutants, we are able to increase micronuclear rupture just by the fact that CHIM7 is in the micronuclei, and even reducing ROS doesn't have anymore any effect. So this means that CHIM7 retention into micronuclei caused their collapse because ROS inhibit micronuclear export of CHIM7. So we put this first puzzle piece in place. To recapitulate this first part, I show you that increasing ROS increases micronuclear rupture, reducing ROS reduces micronuclear rupture, and inhibiting nuclear export also increases micronuclear rupture. But this is true only if the cells are proficient for CHIM7, because if we deplete CHIM7 from these cells, none of these affirmation is true anymore. So CHIM7 is basically the mediator of ROS rupture. But I was left here with a very important question that was unanswered, because CHIM7 doesn't have any other reported function in literature than to catalyze the formation of that complex that we saw before is not involved in rupture. So how can CHIM7, that has virtually no function, collapse a structure like a micronucleus? The answer came again thanks to super resolution microscopy, because I saw that micronuclei with the CHIM7 were more deformed than micronuclei without it. So what I did here was basically analyzing a bunch of these super resolution images, generating a list of vertex for this surface in green, which is lamine, and basically calculating the curvature of the membrane for each of these vertices. And this is basically what I plotted here, the average curvature for each micronucleus. And as you can see, micronuclei with CHIM7 have much more membrane deformation than micronuclei without it. So this, to me, suggested that somehow CHIM7 was interacting with the micronuclear membrane. So I wanted to know which were the binding partner of CHIM7 specifically at micronuclei and whether ROS were changing these. And to do that, we did this very laborious experiment. We purified the micronuclei from the primary nuclei. We lysate the cells, immunoprecipitated CHIM7, and send it to mass spec to see the binding partners. And these are the results. Actually, under ROS condition, indeed, CHIM7 binds more to LMD2, remember the inner nuclear envelope partner, and less the escortry complex. So, based on these results, we built our model that mitochondrial ROS inhibit micronuclear export of CHIM7, but also CHIM7 in the micronucleus binds more strictly to LMD2, which is a structural protein, and polymerizes, forming those big blobs that we saw before in IF. So by doing that, basically pulls LMD2 towards the interior of the micronucleus, deforming the membrane until it breaks. So we set out to basically prove this hypothesis. And since I am a microscopist, if I don't see it, I don't believe it. The first thing that I wanted to know was whether there was truly an interaction between CHIM7 and LMD2. So I used the cells that express CHIM7 fused to a green fluorescent protein and LMD2 fused to a red fluorescent protein. So as you can see, in these cells, we have these micronuclei with these big blobs, but sometimes we also have micronuclei that are the stage before, in which foci of CHIM7 starts to form, right? And if we look at LMD2 in these micronuclei, you can see that it loses this nice nuclear rim localization and it becomes all blobby, exactly like CHIM7. So we knock out LMD2 and we saw the exact same phenotype as we saw for CHIM7, so reduction in micronuclear rupture. And we repeated again that deformation experiment. So as you can see, if the cells don't have LMD2 or CHIM7, they have much less membrane deformation than cells that are proficient for these proteins. So this could have still been done because of these two proteins with two independent mechanisms, right? I still needed to prove that it was the binding between CHIM7 and LMD2 that was causing micronuclear rupture. And to do this, I basically decided to use mutants that cannot bind to each other. So here I'm showing you only the LMD2 mutant that cannot bind to CHIM7, but we validated these results also with CHIM7 mutant that cannot bind to LMD2. As you can see, by using these mutants, we are able to reduce micronuclear rupture independent from the level of ROS. But what is even more important, this rescue of rupture is independent also on the overexpression of that CHIM7 unexportable mutant that before was increasing a lot rupture. So what does it mean? It basically means that it's true that CHIM7 accumulation in micronuclei is what causes their rupture, but only if CHIM7 is able to interact with the micronuclear membrane through LMD2. So we put this other puzzle piece in place. We know that the cause of micronuclear rupture is this increase in binding between CHIM7 and LMD2. We were still missing that part, that polymerization part. So we wanted to know what was happening specifically at those blobs in micronuclei. And we did a nice fluorescence recovery after photobleaching experiment. So for those of you who are not familiar with a FRAP experiment, what we do is basically turning off the fluorescence only in a very confined region, in this case on the blob of the micronucleus, and then study how much time it takes for the fluorescence to come back to the micronucleus. That basically gives us an idea of the exchange between CHIM7 bound, which is often dark, and CHIM7 that is still diffusing, that is fluorescent. So, as you can see, if we do this experiment under control condition, the fluorescence comes back around 30 to 60 seconds. But if we do this experiment under ROS, the fluorescence takes much longer to come back. So this basically means that whatever is keeping the molecules of CHIM7 in these blobs at micronuclei is much stronger under ROS condition. And to understand what is this binding, we did some nice in vitro experiment. So we incubated recombinant CHIM7 with hydrogen peroxide, and then we ran a gel to check the molecular weight. As you can see, ROS super fast induced the formation of this tetrameric form of CHIM7 that again fast is used to form these high molecular weight polymers that are so big that they're not even entering the well. And this high molecular weight form of CHIM7 is completely dependent on the sulfide binding, which is the strongest possible bond between protein. Because if we incubate these high molecular weight samples with DTT, which is a reducing agent, and at this high concentration is able to break the disulfide bond, then we are able to retrieve the monomeric form of CHIM7. So what is happening here? Basically, ROS oxidized CHIM7 cysteines, which are those amino acids that form these bonds. And when cysteines are oxidized, not only they are more prone to form disulfide bonding, but they also instigate a kind of a chain reaction that is completely sequence independent. And this is important because LMD2 has several cysteines that can undergo the same process and form disulfide bonding with CHIM7, while none of the other proteins, escort-3 proteins, have any cysteines in their sequence. So this actually explains that shift in binding pattern that we saw before in the mass spec data. And we put the last puzzle piece in place. So now we know all of the steps through which ROS collapse micronuclei. But we still needed to understand whether this was relevant for human tumors. And to understand this, we use the rationale that actually more than half of human solid tumors is hypoxic, and hypoxia is very well known to increase mitochondrial ROS. So the first thing that I asked was whether hypoxia was able to increase also micronuclear rupture. And to understand this, I basically just cultivated cells into an hypoxic incubator that pumps nitrogen instead of oxygen, so decreases the concentration up to 1%, which is more or less what we see in tumors. As you can see, culturing the cells in this hypoxic incubation for 24 hours is enough to increase micronuclear rupture in a way that is completely dependent on ROS. Because if I feed these cells in the incubator ROS scavenger, then I'm able to rescue this hypoxia-induced micronuclear rupture. So we proved that hypoxia induces rupture through ROS, but also through our mechanism. Because if we knock out CHIM7 and LMD2, we are able again to rescue this hypoxia-induced micronuclear rupture. So the very, very last step was actually going to see what was happening in human tumors, in patients. So we used head and neck cancers that have been classified as normoxic or hypoxic in patients already. And we stained these sections with CGAS, which is another marker of rupture that is often used in pathology. And as you can see, indeed, hypoxic tumor have much more rupture micronuclei than the normoxic ones. So to conclude, I show you that we finally identified the cause of micronuclear rupture, and this is mitochondrial ROS, that inhibit micronuclear export and increase CHIM7 polymerization and binding with LMD2, and also that hypoxia in human tumors is using this pathway to rupture micronuclei. So what we're providing here is one of the molecular mechanisms through which hypoxia drives tumor progression. And with that, I would like to finish. Thank my collaborator, without whom this work wouldn't have been possible. And obviously, thank you all for bearing with me until the end of this talk.
Thank you, Melody. And as mentioned earlier, questions for either right now from the folks that hung on to the end. I'm going to start just since I've wrote down about 4 or 5 questions, but just real quickly. The number of micronuclei in tissue, I know you said the rupture goes up in anoxia, but in the 24 to 48 hours, are there more micronuclei in the first place?
This is a good question, and actually, yes, there is an increase in micronuclei that is dependent on ROS because of DNA damage.
So the damage during, screws up the mitosis, you get more micronuclei.
Yes,
And
yes, exactly.
the micronuclei can be transcriptionally active, I presume?
This
The rupture?
Actually, this is like a very important question that people are like studying right now because there is a heterogeneous population among intact micronuclei. Rupture, no, they're not transcriptionally proficient. But between intact micronuclei, there are some micronuclei that have transcription, and when they do, this is completely skewed towards oncogenes. So it's only prometastatic signaling that is going on there.
Okay. Any questions from the participants in Zoom for Mike or Melody? Nothing in the chat. There was some earlier questions. Mike, you took a look at those earlier.
Yeah.
I know there was a peptide question that I don't think we have anything we can add about peptides in drug discovery. But perhaps, Mike, I'm not sure you emphasized it, but in the space cell painting, maybe comment on the first time it's single cell analysis rather than averaged. And then, the issue of heterogeneity, which is also reported in the new platform.
Yeah. So like Mike said, the two of the other benefits of space over the JUMP consortium cell profiler method are true single cell data where you can go in and look at this cell and get its metrics. But then using Earth Mover's distance, you can also get see how the population, so essentially see how the bell curve moves between different treatments. Does it get thicker? Does it get wider? Does it get thinner? Does it shift left, right? And so one of the, other than just your normal mean, median metrics, you can get these Earth Movers distance metrics as another way of looking at how your population is changing.
Given heterogeneity is one of the classic hallmarks of cancer, being able to report that in these types of screens, I think is a useful endeavor.
I do remember one of the questions from the thing was about looking at organoids, best way to look at organoids in 3D. And if that person's here, I mean, the easiest things are 2-photon microscopy or light sheet, depending on what. Yeah.
Okay, there is a question from Sunita. Sorry if I'm not pronouncing it well. A general comment, is cell painting analysis possible in breast cancer? Certainly, the answer is yes in culture. Mike, you've done some of that, perhaps you can comment.
Yeah, I mean, it's possible in any, I mean, we've done MCF7, we've done the MDA 231s, MCF10A. So in 2D cell culture, it's, I mean, I don't want to blanket, but it's possible to do in any cell type. We are working more towards getting I mean, we've done some work, but looking at possibility of doing 3D cell paint, but that's more then you get into the organoid question that was emailed to us, where how deep can you see into the organoid? How do you keep it going fast, especially if you want to use it for any kind of high throughput screen? But yes, breast cancer cells, any kind of cell line. We've even had it work with like immune cells or cells that aren't necessarily attachment driven.
Good point. A lot of people struggle to work with non-attached cells, but there are ways of getting them to stick and still perform these types of experiments. Back to Melody, I have another one. I know in terms of the rupture, you can see the deformed micronuclei. It's ruptured. You can visibly see that. Is the DNA spooled out into the cytoplasm, or is it just chopped up right away? Do you know?
This is also like a very, very good question. And like sometimes I see protrusion of DNA into the cytoplasm in super resolution, obviously. So like sometimes it's truly spilled and sometimes it just stays there. So like it will actually be interesting to study what is the difference between these two and to actually check whether the spilled one is more immunogenic, for example, in respect to the one that stays inside, and then maybe actually through like find which are the determinants, what determines this phenotype, right? Because we have some idea, maybe there is like this T-Rex one that is involved, which is an isonuclease. So maybe that might be why like it's detached, right? Like it's broken and then it goes into the cytosol. We don't know, but it's interesting. It's definitely interesting.
I'm recalling some really old essays on why nuclei, quote unquote, why do we need nuclei? And some of that early work, more opinion pieces, really was just to keep the DNA from getting tangled up in the cytoskeleton, which leads to another question. I know that the lamina can disappear, as you showed, but what about other nuclear skeleton? I mean, there is an insoluble component, from the biochemistry for sure. Have you done extractions, just to see if your proteins that you're interested in, are they tethered in the micronuclei? Or are they soluble? The FRAP, you can show mobility, but what about a structural component? Is pneuma in the micronuclei? I don't know.
I love that you brought up pneuma because it's one of the proteins that is actually deregulated in the micronuclei. When we do this extraction and we do proteomics and like compare micronuclei with primary nuclei, actually we see a lot less of pneuma and we see less of all of these like nucleoskeleton proteins that are not always dependent on rupture. So like when we do this extraction on the rupture micronuclei or in intact and the rupture micronuclei, we don't see, actually it's always they have this defect in structural proteins. Regarding your solubility question, this is actually like a very good question, and some of my future interest will also go and see like how ROS impact the solubility and diffusion of specific proteins, for example, of the nuclear pore complex. Because I have like preliminary data that show that indeed this is the case. So there is a difference in the tethering and mobility of proteins into the micronucleus, and this seems to be due to ROS. Which are the consequences is still to study.
Given the audience and these general talks like this as part of the IDDD, the drug screening platform that's been around in the Med Center for a while, what do you think of micronuclei in terms of high throughput imaging? I know you're amazing that you did all that manual. I think anybody who's gotten used to some automation to know that you pulled all that off manually is impressive. But there have been a little bit of screening attempt to see what molecules, what functions of known inhibitors affect the number, size of micronuclei.
Yes, actually, I think they're like a very good way and a proxy to screen drugs because they're important for cancer. So we can screen, for example, which an example, we want to see which phosphorylation sites or kinases are involved into this micronuclear rupture. then we actually do like a screen and see under ROS which of the hits inhibits micronuclear rupture through this fluorescence reporter, which is very easy to see. It's like a yes or no, like the one that I use for the XPO. So actually there are several ways in which we can exploit micronuclei and the integrity status of micronuclei to check also whether there will be activation of this interferon or NF-kappa B signaling. So like reporters that study also how this shifts, which is important for the patient response to therapies. So it has important implication in cancer.
Okay, and there's a question from the audience, and I'll read it. It's very brief. Not a scientist, so please forgive me if it sounds basic, but could you expand on the implications that, more directed to Mike, I guess, that this could have on drug discovery and cancer therapeutics? Will this unlock the way to novel therapeutic approaches, more efficient methods, et cetera, et cetera?
I mean, I could also see that being for the micronucleus if there's drug stuff there. But let's see. Can you explain the implications? The implications for me, at least, and is getting, like I said, that mechanism of action knowledge kind of right at the beginning of where it's affecting, how the drug is affecting the cell, how it's affecting the cell population. Is the cancer very heterogeneous, or are all the cells getting killed right away? And to me, that's more in terms of novel therapeutic approaches. One thing that I'm working on now is actually using cell paint to differentiate cell types so that you can mix cell types together, run your assay, and then using machine learning or whatever, figure out which of the cell line is actually, which cells are dying from drug X versus drug Y. So you take a 300 plate screen and shrink it down to a 100 plate screen, which in terms doesn't sound like a huge jump, but as someone who just finished a 256 plate screen, it's very good to get it down as much as possible. But so there's, I mean, already there, that increases your throughput, which makes it more
in general, perhaps a higher level answer to that question is, over the last 10, even 20 years, as the automated microscopy and software has taken off, I mean, it used to be remarkably slow, and most drug companies were just slightly interested in doing anything along these lines because it was not very fast. When, they had, million compounds libraries, they do simple biochemical assays, and, does this bind to that in a multi-wall plate? They can do 1 million compounds really fast. And to do the imaging and analysis, 20 years ago, there was, you could count cells. Well, you could do that easier other ways. But over the years, with the instrumentation getting so much faster, I'm sure we are at least 10 to 20 times faster doing the imaging now than we were 10 years ago with the new instrumentation, which comes with a price, of course, and the same with the software, which is why one of the things with the space platform Mike was talking about, the speed has gotten much better, and you don't need a supercomputer to do it. So that's part of why we really approached putting the time and money into getting that going. Any other questions?
Yeah, there was another one. For the space painting, how can I benchmark or standard, get a benchmark or standard for my cell? So we, between us and the JUMP consortium, there are already a panel of compounds that have specific effects. We in the IMC also have what a small screen called the cell metabolism screen of compounds. It's a single plate, it's 160 compounds, but they range all over the place in their effect, like from microtubule blockers to mitochondria damagers to actin polymerization blockers. So you can also use that to figure out kind of which things affect your cell line of interest the best. I had a question for Melody, if
Sure.
possible. So it's kind of a chicken and egg type question where the hypoxia inside the tumor creates more micronuclei, and then the micronuclei create more aggressive cancers. But is there like maybe a mutation that's the trunk of the tree, and then the micronuclei come later once the hypoxic condition there to make them metastatic or whatever? Or do you think micronuclei are driving from one normal cell to the primary tumor?
That's a very good question, and like I plan to answer this in the future. I have an hypothesis that is that like we know now that the extracellular matrix is stiffer in tumors, and this leads to a different response in the cytoskeleton, which increases ROS production. So I think that this may be the event that starts and then starts this missegregation, and it's like a kind of a cycle that feeds itself, right? So then this will lead to metastasis and even without the hypoxic environment. I think hypoxia just makes it worse and affected the primary nucleus as well. So
it's kind of like you already have bad cancer. This is going to make it worse through micronuclei and okay.
Yes. It's that's what I think it's happening, but it's interesting to study it.
Just one quick other one. Is it ubiquitous? Like, are there any cell lines or cell types that you've come across that are less prone to creating the micronuclei? I don't know for what reason, but do you think that depending on gene expression, there might be some differences to how prone a cell is?
The levels of the proteins you're already looking at.
Yeah, the levels of CHIMP7 or
the level of CHIMP7 is always the same, more or less. It's not correlated with cancer because this is a non-mutational and local process, right?
Yeah.
But it's true that they have like different cell lines have different levels of chromosomal instability in cancer. Normal cells usually don't have micronuclei, so but the cell lines are immortalized, so they already lack these protection. So we don't know.
What I was getting at there is more. So you have like, are breast cancer cells in the tumor more likely to form micronuclei in the hypoxic compartment versus prostate versus lung? Like, have you found anything kind of there?
I see your point. In vivo, no. Actually, like in patients, like almost all of the tumors have micronuclei when they are advanced. So it's very, very pervasive.
Ubiquitous, yeah, okay.
Yeah.
Last question, just real curious. When you said super res, I'm guessing, since I know your background a bit, was that STED?
No, it was not STED. I would have liked it because then the resolution would have been much better. This is a Zeiss with an iris scan microscope detector. So
quasi super res.
Yeah, I mean, it goes down until 120-ish nanometers.
So it's like better than confocal.
It's a little better, yeah. Okay, I think we're kind of coming to the end here. There's no further questions. I mean, we could probably banter back and forth between the three of us, but I think we're wrapping things up. And thank you both. Thank you, those in the audience. Thank you, Dawn, with the GCC helping to set all this up.
Thank you for having me. It was a nice discussion.
Stay in touch.
Bye-bye.